again, I'm Ushma Neill from the Journal of Clinical Investigation with another in our series of conversations with giants in medicine. The control of cell growth was once thought to be passive. When there were cellular building blocks and nutrients, a cell would grow and ultimately divide. But I'm joined today by Dr. Michael Hall from the University of Basel in Switzerland, who upended this simple assumption and instead elucidated the very complex and elegant signaling pathways that result in cell growth. At the core is TOR, the target of rapamycin. I hope today we can learn a little bit about his own growth. So thank you so much for joining me today, Dr. Hall.
Pleasure to be here.
So you have some very international roots. Can you tell me a little bit about where you were born and where you moved and about your parents?
Yes, it's been a rather complicated life history. I was born in Puerto Rico, and my family moved when I was 3 years old to South America, to Peru, where we lived for several years, and then eventually we moved to Venezuela, where we lived several more years, and I eventually left South America to go to the United States to go to boarding school as a teenager.
So what was it that your family did that caused such an international upbringing?
Well, my father was a businessman for an international company. My mother had studied Spanish in college, had a degree in Spanish, and they liked the culture of Latin America, and they decided to make their career in Latin America.
Your mother was a Spanish speaker and your father a businessman, so where did the interest in science come from? Or when did the interest in science emerge?
Yeah, this was a very gradual process for me. I think what set the foundation was my parents instilling in me the importance of knowledge, of education. And I think that came through most clearly when they sent me away to boarding school when I was 13 years old. That was a huge sacrifice for my mother because she was breaking apart the family, but she understood the importance of education. Had I stayed in South America, I would not have received a very good education at that time.
They remained in Venezuela at that point?
They remained in South America or Latin America till the end of their lives, yeah.
So you must have quite a fascinating passport.
I have, I used to, I don't anymore. I have a single passport now. It's a Swiss passport.
You get to boarding school in Massachusetts, if I'm correct.
That was one of these defining experiences in my life, in fact, because it was a huge cultural shock to me. I had a carefree life in South America, in the tropics. Everything was open, free, and then I went to this boarding school, which was at the other end of the spectrum, very disciplined, very rigid. The weather was not the same. I had to wear a coat and tie every day instead of a t-shirt and shorts and flip-flops. It was a radical change for me in every way.
Was it an academic awakening?
It was an academic awakening for me, yes. I think it's there where I really learned to read and write.
So the interest was in reading and writing. How did that then lead you to pursue undergraduate studies in zoology?
Well, that was not a very direct path either. When I was growing up in Venezuela, I did many things. I did a chemistry set, I did electronics, I did art, I did sports. So I was always interested in many different things with no particular focus on anything. And when I went to university, I actually was an art major in the beginning. And I then decided that I needed something that created more of a framework for me. So I decided to study medicine, and that's what I was starting to do, pre-med, and that's why I was doing zoology. I thought that would be appropriate. But then as I was studying pre-med, and at the same time I was working in the emergency room of the hospital, I realized that medicine was not really what appealed to me. So I sort of shifted back more towards art by doing research, where there's perhaps you could say is a compromise between art and medicine.
So interesting that you see the intersection of bench research together with art.
Mm-hmm.
When did you start doing any sort of research?
This started as an undergraduate. I did a undergraduate honors thesis in a laboratory where I first started doing actual experiments at the bench, and I found that incredibly exciting and interesting.
So was it at this point that you thought, all right, I'm done with the MD, not going to do that, but let's think about a PhD, which you carried out in molecular genetics. What made you choose the lab that you choose, and what did you study during your graduate work?
Yeah, at the very end of college, I decided to shift to PhD instead of MD, and I was working in a lab which did molecular genetics, and I was completely fascinated by the kind of science of François Jacob and Jacques Monod, this kind of very elegant bacterial genetics, which is what took me to Harvard, and I worked there with in this area.
You then decided to do postdocs, a short postdoc at the Institut Pasteur, and then headed back to the UCSF. So this is part of your upbringing and your wandering to far-flung locations. What brought you to Pasteur? Was it Jacob and Monod?
Yes, it was. I felt I had to go to, that was the Mecca for me. That was ground zero. I had to go and experience that. I did it. There was also a practical aspect to it because my real postdoc had been planned, and my postdoc advisor, Ira Herskowitz, was in Oregon at the time, and after he accepted me into his lab, he said he was moving to San Francisco. And I didn't want to move to Oregon, and then after a few months move to San Francisco. So I decided I would just meet him in San Francisco instead, and thus I had about 8 months or a year to kill. So I then contacted Maxime Schwartz, with whom I'd already been collaborating as a PhD student, and asked him if I could come to work in his lab for some months, and he said yes, and so I was off to Paris.
So the postdoc with Herskowitz in San Francisco, what did you study with him?
There we studied, there I made a big transition going from bacterial genetics to eukaryotic genetics, in this case yeast genetics, but still the emphasis was on genetics. And I studied there the problem of nuclear protein localization, which was different from what the lab was working on. They were working on signal transduction and mating type control in yeast. This is something that's also marked my pathway. I've always been a bit of a, I guess you could call it a contrarian. I've always been attracted to a problem, but once I'm there, I do something else. And so I started something there which was outside the mainstream of the lab.
Well, I think that came in handy later on in your career, certainly. So what made you decide that you wanted to continue in academia as opposed to a different scientific profession? I
never considered anything else, in fact. And I think this relates back to this, to the importance of knowledge, and this, really thinking knowledge is something to pursue.
So what made you consider moving to Switzerland for your first academic post?
Well, that was not an obvious thing, it's true. I was looking for assistant professor positions in the United States and in French-speaking Europe, because I'd had such a wonderful year in France, and I would like to go back there. And also, what I have not told you is, in the meantime, I'd met my wife when I was in Paris, and she was French, and therefore this made it attractive to go back to at least French-speaking Europe as well. And also, given the fact that I'd always been more global than most, going abroad was not that big of a hurdle like it might have been for some of my fellow postdocs in America who were American and had been in America the whole time. So maybe I was just open to the world a little more than others.
So you speak American English without an accent. Mm-hmm. Clearly you speak Spanish, you speak French. How many languages do you speak?
I speak English and Spanish pretty well. I'm comfortable speaking French. I speak German. I'm not totally comfortable speaking German, but I get by.
Why have you stayed at the University of Basel since 1987?
Well, when I went there, it's a funny story how I got there to begin with, because Basel is not French-speaking Europe, and it's German-speaking Europe, as you know, and Swiss German-speaking, which is not the same, in fact. When I was looking in French-speaking Europe, a colleague in Basel, a future colleague in Basel, heard that I was looking and asked me if I'd be interested in looking at a position in Basel. And in fact, nothing was further from my mind at the time. But I admired him, and he asked me to come look, and I went to see what there was, and I fell in love immediately with the institute where I work, the Biozentrum. I think it's a fantastic institute to work at. I didn't fall in love with Basel immediately, but that came later. It's really a wonderful town. So I've stayed there because the funding and the science is extremely stable, well supported by both local and federal governments. And Basel's a wonderful place to raise a family. And maybe also I was a little tired of moving around so much. I wanted to put down some roots. Basel's an amazing place because it's a town of 200,000. It's headquarters of two major pharma companies. It excels in every domain. Where Roger Federer is from, for example, many famous architects, artists, the list goes on and on.
So let's go back to you starting your lab in 1987. Mm-hmm. What were the projects that you decided to take on at that point?
Well, we were studying as an extension of what I'd worked on as a postdoc. We discovered the nuclear localization signal, which was a bit of a revolution because before that it was thought, and this seems to be a recurrent theme in my research, before that it was thought that proteins passively diffuse into the nucleus, and if they had some role there, which involved binding some substrate, they bound that and they retained there. And if not, they just diffused back out. So there's no active mechanism of transporting something. But that seemed rather naive to me at the time, so I reasoned that there must be a signal in the protein which actively mediates the transport of that protein to the nucleus. And lo and behold, that's what I discovered. There was such a signal, and it's an active transport mechanism. But we did know what this signal interacted with. In other words, what's the machinery that binds this signal and imports the protein into the nucleus. And that's what I started my lab doing, trying to understand what this machinery was. We were making certain assumptions at that time. We thought the machinery would be something in the nuclear pore complex, mm-hmm. and therefore we're focusing all our attention on the nuclear pore complex. But it turns out the machinery that binds, or the components which first recognize the nuclear localization signal, or NLS, are soluble proteins that shuttle back and forth. So we were looking in the wrong place, and as a result, the work was not going very well.
But you had a couple of really great students and postdocs mm-hmm. at the beginning that, through their creativity, helped set you on the right path.
That's right. What really set us on a good path, not the right path in terms of nuclear localization, but a good path, perhaps even a better path, was first Joe Heitman, a postdoc who arrived in the lab from New York. And he was interested in immunosuppressive drugs, which were in those days were quite new, and there was
But why choose your lab to study immunosuppressant drugs?
Well, he came with the idea. He also came with a similar science background I had of bacterial genetics. He was an MD PhD, and the PhD part was bacterial genetics. And he, I think, that's one thing that attracted him, perhaps. I really don't know. And he came to join my lab. He also had to complete his medical studies here in New York, so there was a hard ending to his postdoc. He was newly married, and his wife was a postdoc in Lausanne or Geneva, I think Lausanne, and that was another reason for him to come to Switzerland. So he joined the lab, and he started by working on the problem of nuclear localization. And his project, like everybody else's project in the lab at that time, seemed to be failing because of this faulty assumption we were making. And then he decided to work on these immunosuppressive drugs. So he read the literature very widely, so he knew about these things. Coincidentally, and fortunately, the company which worked on immunosuppression, which was the major company which then had the blockbuster immunosuppressive drug on the market, was Sandoz. So it was a confluence of very fortuitous events. So he wanted to work on these drugs. He had a colleague, someone I already knew at Sandoz. In fact, I was already working with. And so all the pieces were in the right place at the right time, and we got together to start working on these immunosuppressive drugs. We could justify that in the context of what we were studying before. Of course, you have to do this vis-a-vis grants and those kinds of things, because it was known that these drugs blocked intermediate steps in signal transduction, in T cells in particular. That's why they're immunosuppressive. And in those days, no complete signaling pathway was known. Now we know many.
We see the spaghetti of all of the
exactly.
signals,
Now,
right?
The explosion of knowledge in this area over the last 30 years is incredible. But back those days, relatively few pathways were known, and no complete pathway was known. And this step between a receptor being activated on the surface of a cell, for example, the antigen receptor on T cells, and the transcription factors which are then activated in the nucleus, this step in between, which was a signal, whatever that was. Nobody even knew what that might be. Protein, small molecule of some other nature. How that was transmitted from the receptor to the transcription of the nucleus was unknown. And of course, this step involved going from cytoplasm to nucleus. So it fit in a very broad context what we were already doing. So I could justify it in terms of our ongoing research.
So how did you justify doing this in yeast instead of in T cells?
Well, this is actually what I found very exciting. I mean, it's needless to say, everybody else was working with mammalian cells. These were drugs. Cyclosporin was already on the market. Two other drugs, FK506, Rapamycin, were still being developed. And they worked in the clinic, but nobody knew how they worked. And everybody was working on mammalian cells, of course, because they were drugs to be used in humans. And Joe and our collaborator at the time, Rapamycin, who was at Sandoz, who was involved in the immunosuppression work at Sandoz, had the idea to use yeast. And this seemed like an outlandish idea to many, but what many did not realize that these drugs were actually natural compounds which evolved as weapons in microbiological warfare. In other words, they evolved to act on other microbes such as yeast. So what Joe and Rao decided to do was just to use these drugs for what they evolved to do. And maybe if you look at it from that perspective, the notion of giving these drugs to humans is somewhat outlandish.
So you said rapamycin for the first time.
Mm-hmm.
When Rao decided to give you some rapamycin
Mm-hmm.
together with all of the other immunosuppressants,
Mm-hmm.
did you have any idea that it would be such a key part of the rest of your career?
No, I had no idea. I had no idea. These drugs were creating lots of excitement in these days. They were relatively new. They were responsible for medical breakthrough. Transplantation surgery became a clinical procedure, whereas it had been an experimental procedure before. So there was a lot of excitement about these drugs. And understanding how they worked and what they bound was viewed as an important step. And it was extremely, extremely competitive to make that step. There were many labs working on it. Of course, everybody else was working with mammalian cells, and we were able to win the race with regard to rapamycin, I think because we worked with yeast.
So you originally found that there's TOR1 and TOR2, and then later went on to isolate the two different TOR complexes.
Mm-hmm.
Originally, though, during this time, you had thought, given that when you knocked it out, the cells didn't divide, that
Mm-hmm.
TOR and the target of rapamycin were essential for cell division.
Mm-hmm.
Was it then a gradual understanding that it was more about cell growth, or did you have an aha moment, like a face slap aha moment?
Yes, this is when we made you were asking earlier whether I knew it would turn into something important. So it was important on the short term. We knew this because we were elucidating how these important drugs are working. And to tell the truth, the project almost died at that stage in the lab because we'd answered this initial question which we set out to answer. And Joe left, and the mutants, the TOR mutants that he'd isolated, were sitting in the freezer. And then another student's project fell through, so she picked up the mutants, started characterizing them, cloning the genes and sequencing them. And when she knocked those out, the cells arrested in the G1 phase of the cell cycle. And that's the same effect rapamycin had on yeast cells as on T cells. So this is the single most important phenotype we had, was this cell cycle arrest. And also in the context of those days, the cell cycle was a very, very active area of research, and very exciting. This is what you read about everywhere. We were excited because we thought we had a new angle to approach the cell cycle via these mutants and rapamycin. And we thought we had perhaps discovered a new control mechanism for the cell cycle. And this is why, in fact, partly explains why Joe picked the name TOR for these mutants. But we can come back to that later if you'd like. So we, for a number of years, were working with this assumption that TOR controlled the cell cycle, progression through the cell cycle, and cell division, which is the process which leads to an increase in cell number. And it was actually a rather dark phase in the lab because all our experiments were based on that assumption, but they were not giving the answers that we hoped or expected. And then how did we make this transition to, was it an aha moment or gradual? I would say it was gradual with a few mini aha moments sprinkled throughout. I think one of the first important mini aha moments was a seminar I gave in Vienna. And I was invited there by Kim Naismith, who was a cell cycle expert. And I presented our data from the perspective that what we have here is a system to control the cell cycle, cell division, increase in cell number. And they were experts, long-term experts in cell division. And what I described did not fit with what they knew and what they were working on. So they were very skeptical. perhaps they felt that if it was playing a role, they would have come across it, but they hadn't. So they were the first to plant this seed of doubt that maybe it's not cell division we're studying, but some other process. And then we started wondering, well, if it's not cell division, what could it be? And this actually was a discussion that followed after the seminar during the question and answer session in Vienna. And we were discussing, and we eventually came to the possibility it might be controlling cell growth, increase in cell mass or size, as opposed to increase in cell number. And this was a mini revolution in itself, because cell growth was, as you mentioned earlier, was not thought to be a regulated process, thought to be a passive process. So therefore it was not thought that it had a regulatory system behind it, which is why we never considered cell growth. And, but now, after a process of elimination, we then had to start challenging our assumptions and the existing paradigm, or the conventional wisdom. And maybe cell growth is controlled. And if it's controlled, maybe then that is what TOR is doing. And once we had once this switch was flicked in this direction, then we could start looking at all our data in a different light. We could start designing experiments in a different way to test a new hypothesis. And then everything fell into place immediately. Everything started to make sense. And then for about a 10-year period, we were in a kind of a research paradise, because we stumbled into a new field. And almost we could look under any rock and find something interesting and important. It just came from having this changing our assumptions as to what was happening.
So your research has pivoted then in the more recent years to more mammalian cells and less yeast, because of the amazing translational promise of rapamycin in cancer and diabetes and in any number of different things. Are you happy about that, or are you happiest when you're back in your yeast genetics?
I am happy in both. We switched to mammalian work when did we do this? About 15 years ago. Just at the point where you could start doing genetics with mammalian cells, where knockdown technology came into existence. And remember, I'm at heart a geneticist, and this is what held me back from working on anything above yeast. And then once it became possible to do genetics in mammalian cells and tissue culture, then we started working on mammalian cells. And of course, the first thing we wanted to do was to see was whether what we discovered in yeast was conserved all the way to human. And then for many years, the lab playbook was to use yeast as a roadmap to make first discoveries or get glimpses of what the biology might be. And working in yeast, you could do this relatively easily, and then test what we'd found in yeast in mammalian cells. And almost invariably, we saw that what we discovered in yeast also applied to mammalian cells.
So very conserved pathway. Very conserved pathway, yes. How many times a day do you think you say the words TOR or rapamycin?
Several. I don't count it. You know, one of the criteria I have for picking a name of a gene or a protein, we've discovered many genes. As a geneticist, we've discovered many genes. I had a rule in the lab that to give a name to a gene, it has to be something you can say quickly 10 times in a row. It has to be able to flow off the tongue very easily, because if you're giving a seminar, you have to be able to say it. And also, if you can say it easily, you're going to be able to remember it easily. And TOR fit this perfectly.
How did you come up with the name TOR? Or how did Joe come up with the name TOR?
We actually went back to look at his lab notebook, and he had there's a page, it's actually December 2nd, 1990, where he wrote name, question mark, and then listed all these possibilities. In yeast, genes and proteins, there's a specific nomenclature convention, and it's a 3-letter acronym. So we are confined to 3 letters. And he was making a list of all these three-letter names based on rapamycin or FKBP, which is the protein that rapamycin binds to then bind TOR. And you had PIF, PAF, RAT, TFR. And TOR was somewhat down the list. And I liked TOR. It was just it's very easy to remember. It's a very common set of three letters in the English language, actually. So it's already in our minds. It was a natural name. Joe had the idea of TOR. He liked TOR because he lived near the lab, near a gate, or the old gates, the medieval gates that went into the city, called Spallentor. And Tor in German means gate, so Spallen, gate. And this is the time we thought Tor controls the cell cycle, the circle of the cycle of division. And the Basel, having once been a fortified city, had a wall around it, and this gate was one of the gates to enter the city. And he thought very romantically that the Tor was the entry into the cell cycle, much like these gates were. So he thought this was a terrific name. It also reminded him of Spallentor. Probably to this day he thinks of Spallentor when he
But Tor also means something else in German.
Yeah, Tor has two different meanings depending on what gender. Gate is das Tor, which is the neutral word. But the masculine form of Tor in German, der Tor, means the fool. But Joe wasn't deterred by that.
Foolish gate,
Yeah.
I tell you.
I was more worried foolish model, but.
Do you use a lot of Tor puns like caveat emptor and torque and story?
I used to, much less now. Caveat emptor I've never used. I've used torque and Tor story and things like this, yes. Yeah. When I go to Toronto, I make jokes too. But.
So Rapamycin gets its name from Easter Island, which is called Rapa Nui. Have you ever been?
I've never been, no. I talk about going often, but one day I'll go, yeah.
Well, perhaps we're here today in town, or you're in New York City to accept the Lasker Award, which is the latest in many of these awards. Maybe you can use the prize money to go to Rapa Nui, visit the home. But what have these awards, the Gardner Award, the Breakthrough Prize, now the Lasker Award in Basic Science, meant to you? And does it mean your family understands a little bit more why you spend long hours in the lab?
There's, of course, a huge amount of gratification in winning these awards. It's like someone pays you a huge compliment, and everybody loves to receive a compliment. So you of course, feel very good about it. But it also is validation of what you've spent your life doing, which is also very gratifying. But has it fundamentally changed me in any way? I hope not. I don't think so. My family tends to be rather casual about these things. It doesn't. Nobody seems to be very impressed around my house. Yeah.
If you weren't able to be a scientist, what else do you think that you would have done in your life?
It certainly would have been something where there's a creative element, much like science. And maybe this gets back to what we were discussing before. Maybe I would have been an artist. I don't think I had the courage to be an artist because it's too free-form. Science allows this creative element, but in a more structured environment. And that's maybe why I became a scientist. But maybe if I'd had the courage, I would have been an artist.
Do you still do any art?
No, I stopped quite a long time ago, but just for lack of time. But hopefully one day I'll get back to it.
It has been a real pleasure hearing a little bit more about your growth pathway. Thank you so much, and congratulations again on the Lasker Award.
Thank you, Ushma Neill.